In particular, I knew Ivano over a period of 20, probably more years. Ivano was a pioneer in our field, and to receive an award that carries his name makes it even more worthwhile. I'd like to thank all of the people involved in the nomination and evaluation process. I'm going to talk to you, as Shoras has just beautifully introduced, I'd like to thank you for that beautiful introduction, about how we use NMR spectroscopy to study the role, and we have done for the last 20 years, 20, 30 years, to study the role of intrinsic disorder in functional biology. Okay, sorry. Simultaneously to the revolutionary advances which have put structural biology into an entirely new place forever, there was a gradual realization that, as Shoras has just said, a significant fraction of all proteins that we are aware of actually code for regions which cannot really be studied using solid-state structural techniques, shall we say. And of course I'm talking about intrinsically disordered proteins. And I'm also going to talk to you particularly about the role of NMR spectroscopy, which is particularly appropriate precisely because it provides site-specific ensemble and time-average structural parameters from throughout the protein. In relatively good quality spectra where we can study and we can measure a lot of parameters. So how do we do this? Well, we, and many of us, have spent a lot of time thinking about how you represent an intrinsically disordered protein. And we developed methods of ensemble descriptions of the intrinsically disordered states available to the unfolded protein, exploiting a highly efficient conformational sampling algorithm, a good deal of NMR theory, which is on the shoulders of giants here, fortunately in this field. A specifically designed genetic algorithm in our case to develop representative ensembles in agreement with the experimental data, which then allow us to delineate the contours of conformational space, which are sampled by the protein under those conditions in solution. And of course, due to the astronomical number of degrees of freedom available to such a system, the emphasis, of course, always needs to be on calibration, cross-validation, and evaluation of the predictive accuracy of the methodology. I won't go into these details. Now, in order to study interactions, NMR is very powerful at studying interactions. I'm sure many of you are aware. You can titrate your partner protein into your IDP of interest, and whether you're in, depending on the exchange regime between the different exchanging species, if you're in fast exchange, you can do chemical shifts and relaxation parameters, and if you're in the so-called intermediate exchange, where your peaks get a little bit broader, then you can measure so-called chemical exchange methodologies. And these give you really a lot of information. They give you information about populations, about the structures of the exchanging species and the exchange rates. And to illustrate how powerful this can be, we measured carbon-13, nitrogen-15 and proton relaxation dispersion from throughout an intrinsically disordered protein, and combining all of this information together, we could map the interaction trajectory of this IDP from the free state equilibrium. Of course, we always need to characterize this to show how this is conformationally funneled via an encounter complex into a specific binding pose on the surface of the protein. Now, not all intrinsically disordered proteins fold upon binding. In a collaboration with Lemke many years ago, mediated by Sigrid Milles, we studied these long disordered nuclear poring chains that actually fill the nuclear pore. This looks empty, but it's not, it's actually full. And these are decorated with so-called repeat motifs, FG repeat motifs. This is about as short as you can get for a motif, which are responsible for specificity and transport efficiency. And using NMR spectroscopy, we were able to demonstrate that the intrinsic affinity of each one of these short motifs, which is responsible for the specificity of binding the transport, is about 10 millimole. But the nuclear pore is full of these motifs, and therefore this provides a polyvalent reactive complex, which, with fast on and off rates, is responsible for the fast transport. And in particular, NMR shows you that looking at the backbone conformation of these binding sites, their conformation doesn't change between the free and the bound form. So they really don't fold upon binding. These are side chain interactions. So there's obviously a great variety between all of these different types of intrinsically disordered proteins. One thing that holds them all together is that they are all dynamic. And although the conformational space of IDPs is quite well understood, a lot less, it's much more challenging to say something quantitative about their dynamics. Despite the fact that NMR relaxation, which we measure in every NMR study of an intrinsically disordered protein, actually directly reports on IDP dynamics in the picosecond to some number of nanosecond time scale. And we wanted to know whether we could develop a physical framework to interpret these relaxation rates. I won't bore you with the detail, but we measure relaxation rates which are dependent on timescales. The different timescales we measure them at different magnetic fields to extend this range. And we measure these as a function of temperature. And on the basis of this, interpreting all of these data together, we were able to identify 3 distinct contributions to the relaxation that we measured, reporting on vibrational motions in tens of picosecond timescales, maybe these are of less interest, backbone dihedral angle dynamics, which of course are of interest, so this is local structure, and chain-like segmental motions, which of course can also be functionally important. Now, IDPs evolve in highly complex environments, so we wanted to know whether we could incorporate the complexity of the physiological environment into our analysis. So we do the same kind of analysis here, but this time as a function of different Crowders, some kind of nano-viscosity that we're changing over a function of the experiment. And on the basis of this, we can write down relatively simple expressions which allow us to correlate the timescales of the motions with the viscosity of the surrounding medium. And this, basically, the surrounding environment. This, therefore, allows us to reveal sequence-dependent coupling of local and segmental motions to this local environmental viscosity. This turns out to be extremely robust and transferable, even predictive, between different experimental conditions, for example, provided by different viscogens, as well as rationalizing and allows us to predict quite accurately relaxation, which is measured in live cells where IDPs have been injected and measured over a period of 48 hours. So this provides a framework for interpreting NMR relaxation in very complex environments. Okay, now in order to go further in time-honored tradition, we combine NMR measurements with molecular dynamics simulation. And here we developed an approach which actually mimics precisely the experimental averaging process by describing the dynamics and the NMR relaxation that we measure as an ensemble of time-dependent trajectories. This is no longer an ensemble to describe the free energy landscape. This is also describing the kinetics of the system, so the timescales which are involved. This turns out to be nicely robust and very predictive of relaxation rates which are not used in the active selection process. So we're quite happy about this. And with this in hand, we can then develop analytical motional models which can be used for every amino acid along a sequence, and we can identify what the physical nature of the motion is, deconvoluting different contributions, and to identify, for example, correlated segmental motions along the sequence of the protein. We can even use this kind of approach to, in a kind of forensic way, to try and figure out what's wrong with molecular dynamics force fields which are currently available, which actually don't predict very well relaxation or dynamic properties of intrinsically disordered proteins. And by comparing simulation and experiments and a motional model, we can identify what what's wrong with these force fields and what needs to be amended. Okay, and so we'd like to apply these methods to even more complex environments. And of course, liquid-liquid phase separation is something which is very important and is a very, very different type of environment than what we're normally used to studying when we're studying an isolated protein in the dilute phase. Here we're looking, it's kind of a model system. It's the C-terminal domain of measles virus nuclear protein, which under certain conditions phase separates. We have one phase in the NMR magnet, and we can look at the nature of the system using NMR spectroscopy. On the basis of our NMR measurements, we can see that the local free energy landscape is not really modulated by phase separation. But the dynamic properties are massively changed. And by combining NMR relaxation with exascale MD simulations of liquid-liquid phase separation-like conditions, looking at the effect of hypercrowding, self-crowding of the protein, we're able to identify what these differences are, which include a slower timescale of all modes and a significant redistribution of these modes relative to the modes which are present in the dilute phase. Okay, I won't go through any more detail. This is a little bit more technical. And I'd like to just finish looking at a couple of examples where we've used these methodologies to try and understand the role of disorder in viral replication complexes, in particular the viruses, RNA viruses, which are important for human health. The first of these is measles and associated paramyxoviruses. And in particular, we're looking at the complex formed by the nucleoprotein and the phosphoprotein. This is actually a chaperone complex, which keeps the nucleoprotein from binding non-specific RNA prior to encapsulation of the viral genome, where, of course, then it provides this protective sheath around the RNA. This is highly dynamic. The tetrameric phosphoprotein has intrinsically disordered domains from 300 to 500 amino acids, depending on the paramyxovirus. And when you consider that, for example, measles has only 16,000 bases in its genome, and yet it requires that 300 amino acids are intrinsically disordered, then they are probably doing something. It's parsimonious, it must be doing something. But we don't really know what this is. So we studied this complex. So this contains the entire disordered domain of the phosphoprotein and the nucleoprotein with its disordered domain. This is the spectrum of this complex. It actually comprises 450 intrinsically disordered amino acids, but they're all pretty much resolvable, and we can see what's going on in this complex. And actually what we discover is that in addition to the putative primary interaction site, which is the first two N-terminal helices of the disordered domain, which bind the nucleoprotein, there is actually a second interaction involving a transiently populated helical domain, which is very short motif, which is 150 amino acids downstream, has an affinity for this distal part of the nucleoprotein, which is 5 orders of magnitude weaker than this one. They can both be detected, by the way, using the same experiments by NMR, which is something, again, quite remarkable for NMR. This is 5 orders of magnitude weaker and associates and dissociates on and off the surface of the nucleoprotein. Then you're going to tell me, okay, if it's so weak, it can't be physiologically important. If you mutate these 4 residues, then it goes to hell. First of all, the interaction disappears, but in mini-replicon assays, there is no more transcription or replication. So this interaction is actually essential, and is essential to the nature of the chaperone complex. Okay, these two proteins have actually been known for about 30 years using different techniques to co-localize in vivo, forming puncta in infected cells. And actually, it did occur to us that this might be to do with phase separation. We could demonstrate that this is actually the case. When you mix these two proteins, they do phase separate. And using either fluorescence imaging, fluorescence spectroscopy, and NMR spectroscopy to look at the role of the intrinsically disordered domains in the stabilization of these droplets, we were able to characterize this and even to demonstrate that when RNA is added to the solution, it's taken up into these droplets, and nucleocapsid assembly occurs within the droplets and is, of course, accelerated because the concentration of the components within these droplets is obviously very high. Now, we think that this is part of the much vaunted viral replication factory machinery. It might not be making the car, but it's maybe putting the door on the car, but it's part of the factory. We use the same kind of approaches, I won't go into any details here, for RNA viruses which have become more important over the last few years, in particular looking at the nucleoprotein of SARS-CoV-2. This is, again, the most abundant protein in the virus. It's responsible for RNA genome encapsulation, and its central disordered domain comprises mutations which are important for genome replication that define variants of concern, for example, Delta and Omicron. So this clearly plays some important role. And we were able to show that this binds the N-terminal domain of NSP3, which is its viral partner protein, and via this interaction, the intrinsically disordered domain folds around the surface of the N-terminal domain of NSP3, and this results in a massive compaction
and Sachs was also very important to demonstrate this, a massive compaction of the conformational space available to the entire full-length nuclear protein, and that this modulates its ability to bind RNA. So again, maybe some kind of chaperone complex. Okay, and finally, for many years we've been studying with our colleagues in Grenoble, including Darren Hart, who's here in the room, the molecular basis of adaptation of avian influenza to humans, which in fact just seems to be getting more and more important as time goes on, and couldn't be any more important now than it actually is. So it's been known for many years that adaptive mutations for replication in human cells are concentrated in the polymerase PB2 segment in its C-terminal 627 NLS domains, these two domains here. In particular, the E627K mutation is essential for H5N1 replication in human cells, and this is therefore concentrated a massive amount of attention in many groups throughout the world. We demonstrated that this is how it crystallizes, these two domains. We demonstrated using NMR spectroscopy that there is actually an exchange between an open and closed form using NMR single molecule FRET and small angle X-ray scattering. We could show that there was an open and closed equilibrium between the crystal structure and an open form. And in fact, this open form is essential for binding importin to import the PB2 segment into the nucleus, which is where replication occurs in influenza. Things became a great deal more interesting in 2016, when Wendy Barclay's lab at Imperial College identified AMP32A, a key host transcription factor that appears to be responsible for this host adaptation. AMP32A has a folded domain and an intrinsically disordered, highly acidic disordered domain, which in birds is 33 amino acids longer than it is in humans. And this 33 amino acid deletion in humans comprises 27 amino acid repeats of the previous 27 amino acids and a hexapeptide, a 6 amino acid hexapeptide that is avian unique. Remarkably, avian AMP32 rescues activity of avian polymerase containing this residue 627E at the 627 position in human cells. Similarly, human polymerase, that means with a K here, requires the presence of AMP32, the human form, the shorter form. So the question then is, how does one adaptive mutation, E627 to K, compensate a 33 amino acid deletion in this AMP32 IDR? So this is clearly a complex problem. We have an intrinsically disordered domain which is interacting with a protein which has an open and closed equilibrium. So the first challenge, therefore, is to develop and compare atomic resolution models of these two complexes. First of all, we should see if they interact. Well, if we look at AMP32, we indeed see an interaction when we add the two polymerase domains. The interaction is distributed along 60 amino acids of the human IDR, and in the avian case, it involves this avian unique hexapeptide. Crucially, the affinity is weak in all cases, again, between 600 and 1500 micromolar. We look at the interaction from the side of the polymerase domains, which of course we can also do. Here we see that indeed human AMP32, this basic protein, interacts with one of the basic faces of the 627 domain. But the affinity here, measured from this side, is very different. It's 20 micromolar instead of 600, suggesting the presence or the existence of an electrostatically driven multivalency of the IDR for this basic surface. Because the affinity is so high, is so weak, measured from the IDR, all of these different IDRs are interacting with the same surface in the polymerase domain. Now, in order to characterize this in a little bit more detail, we make 16 different forms of our 627 NLS. In each one of either the human or the avian, we mutate one residue and we add a paramagnetic probe. And then we measure intensity, paramagnetic relaxation and enhancements of the partner when we titrate this into this solution. And these are shown here, a comparison of the human-human complex and the avian-avian complex. And we use these as primary data to develop ensemble descriptions of these two complexes. Now, looking at them like this, you can't see anything, so we need to be able to compare them somehow. And the way we do that is to look at a difference-distance matrix, which basically compares the distances between the common intrinsically disordered region, these first 100 amino acids, and the different regions of the polymerase domains. And what we very clearly see, so blue basically tells you where the human-human form are closer, and red tells you where the avian-avian form are closer. And what this basically says is the human adaptive complex exploits many different multivalent electrostatic interactions between the acidic tail and this basic surface. It is, of course, enhanced by the presence of 627K, and this basic surface is interrupted by 627E. And in this case, the interaction is rather mediated by this hexapeptide with the second NLS domain, which is all very fascinating and a bit of a challenge. But we still don't understand the driving forces behind adaptation. And we wondered, were we missing something? In fact, other people in the field as well, the Oxford group of Jonathan Grimes and Fodor also came to very similar conclusions. We were missing something. Remember, in measles virus, the phosphoprotein chaperones the nuclear protein prior to RNA encapsulation. Similarly, SARS-CoV-2, the disordered nuclear protein, folds around its viral partner in an apparent chaperone interaction. The influenza, Northern myxoviridae in general, do not express a phosphoprotein or a protein which can actually carry out this feature. And it did occur to us that maybe AMP32 was being hijacked by the virus to play the role of the paramyxoviral P protein. In other words, it would interact with the nuclear protein, as well as the polymerase. And this turns out indeed to be the case, where we do see an interaction, and remarkably, the avian influenza, the avian AMP32 interacts 33 amino acids, precisely the length of the deletion between the two forms, further along the intrinsically disordered domain. There are slightly different affinities. In the case of the avian case, we can very clearly show by NMR that the AMP32 binds both the polymerase domains and the nuclear protein via distinct linear motifs along the disordered chain. So this makes sense, and it seems to be acting like a hook protein that could, of course, co-localize the nuclear protein and polymerase to encapsulate very efficiently the emerging newly synthesized RNA. In the human form, things are very, very different. So we measure relaxation dispersion. In this case, we're in an exchange regime which requires that we do this. This gives us two important observations. Firstly, the effective local KD for each of these different amino acids in the unfolded chain is about 50 micromolar. Remember, the ITC-derived number was 1.7 micromolar. So again, we seem to have electrostatically driven multivalency with respect to the nuclear protein. But more remarkably, the chemical shift perturbation induced by binding the nuclear protein is essentially indistinguishable from the chemical shift perturbation derived from binding the polymerase. So are they in competition? Well, it turns out they're not in competition. NMR very clearly shows that rather than competing, human AMP32A simultaneously and multivalently binds both the polymerase domains and the nuclear protein via the identical extended motif. This multivalency is no longer multivalency between different sites on the surface of one protein. It's multivalency between different sites on the surface of one protein and different sites on the surface of the other protein. It's keeping those two together in a ternary complex. Okay, so this is summarized here. So the host AMP32 is indeed a proxy phosphoprotein for flu virus. AMP32A, avian AMP32, rather co-localizes polymerase and nuclear protein via distinct binding sites, providing an efficient mechanism for encapsulation of the newly synthesized genome. These two binding sites collapse in human AMP32. In response, the virus mutates this amino acid, which creates a more continuous electrostatic surface, promoting multivalency with AMP32A, such that a multivalent ternary complex can be formed, which mediates the interaction via the same 50 amino acids between the two viral proteins. Now, this demonstrates firstly the importance of the previously observed multivalent interaction, which is unique to the human adaptive polymerase, and apparently, finally, reveals the molecular basis of this crucial adaptation. Never underestimate viruses, they're very clever. So this is just the last slide, which is just a little bit of context. And context is essential. I haven't really talked about context, but we don't understand anything about how IDPs work unless we have the molecular context, which is provided very often these days by electron microscopy or by X-ray crystallography. This is a structure actually of influenza C polymerase determined by the Fodor lab. Now there are structures of A and B which is sufficiently similar for this comparison to hold. It turns out that this functions as a dimer. One of these is the replicase, so the RNA comes out of this. It's tethered in the encapsidase until enough of it is out so that it can bind the nuclear protein. These are these two 627 domains on the surface of this. We can speculate about where the intrinsically disordered protein could go. I should say that this is bound to AMP32 folded domain, but of course you don't see any of this folded domain significantly in the EM structure. But we can look at this using solution state NMR, because if this remains flexible, then we can see, in fact, this is actually a spectrum of 8 micromolar that took 48 hours, but you actually get spectra which very clearly show the validity of the characterization of the system using our reduced system. We see the same chemical shifts induced. So this is still interacting with these two 627 domains. And so we can then retain those conformations which are able to give rise to these chemical shifts, and then we can speculate about the position of NP on the basis of our characterization of the ternary complex to propose that AMP32 acts as some kind of molecular glue between these two basically charged proteins and a negatively charged intrinsically disordered domain, which co-localizes these two so that this can then bind the emergent RNA. Okay, so I finished. Okay, I want to go through a lot of people. I've been extremely lucky in my group to have some extremely talented people. I think maybe I should start with the second bottom line. These 5 people who were either PhDs or postdocs, most of them postdocs, but Loic was also a PhD student. 4 of these now have ERCs and their own labs, and one of them is head of biomolecular NMR in France for Sanofi. So I think they've all gone on to do great work. But while they were in the lab, they were brilliant and they did brilliant work, and I profited from this as much as the field. The current lab members, I'd like to mention Aldo Camacho-Zarco and Lithan Yu for the flu work, Serafima Guzeva for the measles work, and her work with Vincent Schnapka for the work on the droplets and the MD simulation of the droplets. Louisa and Emmy and various other people for the SARS-CoV-2 work. The rest of the names are here. I'd like to thank you for your attention. I'd like to thank again the nominators and the evaluation committee, and I'm open for questions.